Good afternoon, Professor Kagan, Dr. Yumi, Dr. Badmos, and all the participants present. Okay, so I'll be presenting chemical-induced models of cancer in Drosophila melanogaster. The presentation session is going to be for about 30 minutes, and then we'll go to the lab to experiment it. Is that okay? So it's important for us to be able to know how you can use chemical approach to induce different diseases. I will also start with my journey in Drosophila research. I'm sure some people were like me when I started, that they did not know anything about the fly. And then they are now afraid, thinking that, how will I be able to embark on this journey? I want you to know that I was also at a time like that. And then I was able to adjust. I didn't know anything about the fly. Apart from in biology, you remember in biology, when you cross white eye fly, red eye fly, that was the only thing I remember about the fly. But I contacted Professor J.V.T. Rocha that I would like to visit his lab for a one-year postdoctoral training. And he told me he already stopped working with rats because it will take up to 6 months to get ethical clearance before you work with rats. And I had just 1 year. And you can imagine if I use 6 months to seek ethical approval, where will I have time to do the actual research? So I wrote the proposal and he corrected it, and eventually I got the postdoctoral award, which was actually first CMPK postdoctoral fellowship. And then after the 1 year training, I returned to Nigeria with 2 vials of flies. I checked in the 2 vials of fly in my bag, and then I returned to my department and I started work with the fly. So this was my office, the office given to me, and I turned the office into a mini fly lab. That was 2014 to 2017. I used this place, supervising students using the same table. They'll be working on the flies, banging the table while I work on the same table in my office. And then after 2017, I was given a particular space. We started with this space, and then after some time we got the fly lab, the fly up. So we are using a whole building now for the study building as our fly up, where we breed the flies. And then we also have the analytical lab, the molecular lab that we are using at the University of Ibadan. So due to demands for training, because after different workshops, there was increase in the demand for training, and I have students to supervise, teaching loads, and several other things. That inspired us to commence the Drosophila Research and Training Center, where we are currently, from 2 vials of flies to the Drosophila Laboratory and a research center. So this is the auditorium, and then we have the laboratories just opposite us. And I don't know if you can move the laptop closer. So my area of focus as a researcher, are you changing it? So my area of focus is to understand how different environmental contaminants, environmental toxins, predispose to different diseases, associated with neurodegenerative diseases. And then, these environmental toxins can actually predispose to different diseases. But my area of interest is those that can predispose to neurodegenerative diseases and also cancer. And this will enable us to be able to understand the strategy to put in place for prevention and also possible therapy. By collaborating with different researchers like Professor Kagan, Dr. Alex, I also work with flies that are actually genetically worked upon to be able to study specific diseases. One disadvantage of working with chemicals is the fact that it can attack different systems. It's not specific. For example, a neurotoxin can also have adverse effects on the liver and other organs. So that's just the disadvantage of working with the chemicals. So, but it's also something that was useful for me when I started, because I did not do anything about fly genetics when I started working with the fly. So a brief introduction to Drosophila melanogaster, like the participant that came from Unilag said, she just asked about fly, and some other people, maybe this is your first time working with flies. So I will just introduce it a bit. About 75% of disease-causing genes in human are conserved in the flies. Apart from that also, the entire genome of the fly has been characterized. So we understand all the number of genes present in the flies, all the proteins, everything has been understood. And that of human also has also been characterized. As a result of that, they now found out that diseases that are caused as a result of genetic abnormality, 75% of those genes are conserved in the flies. And about 60% of the genes that humans also have are also present in the fly. And also, apart from that, it's also very easy to manipulate based on what Dr. Badmos has been talking about in the last session. They also have a very short life cycle, approximately about 10 days. And that's something that is very important if you compare with that of rats. We have the laptop here. So like I said, that Drosophila has a well-characterized genome, about 50,500 genes on 4 chromosomes, compared with humans where we have 22,000 genes with 23 chromosomes. And this small genome, 3 chromosomes, and high gene conservation actually make the fly relevant for studies, for research, environmental research. It's about, with respect to the study of human diseases, we have different diseases that have been studied using the fly. For example, we have different neurodegenerative diseases, infectious diseases, metabolic diseases, cardiovascular diseases, genetic and developmental disorder. In case yours is not cancer, there are different diseases that you can use the fly to actually study. This is important because some people will be looking at the morphology of the fly being a very small animal, and then you look at the morphology of man, that there is no comparison. If you look at that based on morphology, you will assume that it's not actually comparable. But if you look at it based on molecular processes that actually govern different diseases, you will see similarity with that of human. One important thing I like about the fly is that there is no developmental stage of the fly that you cannot use as a model. For example, if I'm interested in developmental toxicology, we can use the embryo of the egg. We can use the larva to study organisms that grow. At that stage, whatever you give them, they eat. Especially if you are working with drugs that are toxic, if you expose the larva to the drug, they will eat it. If you expose an adult fly to something that is toxic and they eat it and they see that it's toxic, they will try to avoid it. Although there's another approach that you can use to expose them to such drug by maybe if you coat it with a sweetener. You coat children medication with something that will make the taste bud to feel like taking it. But the larva stage is a very good stage that you can use for study. The pupa stage and even the adult stage. So there is no stage of the fly that you cannot use to study. What about with respect to mammalian-like intestinal system? The fly has homology with, for example, the salivary gland. The fly has salivary gland, the fly has esophagus, they have stomach. They are insulin producing cells. They don't even have liver, but we don't call it liver in fly, we refer to it as fat body. So the different organs that we have in our system, the fly also has something similar that carries that function. Nobel Prizes that have been awarded based on studies on flies. It's interesting to know that from 1933 to 2017, 6 Nobel Prizes have been awarded. The most recent one is the one on circadian rhythm, awarded to 3 scientists, Professor Jeffrey, Michael, and Professor Young. And the 3 of them were awarded £825,000. They share that based on the study on the circadian rhythm, from heredity to influence of mutation of genes, odor receptors, innate immunity. These are studies carried out in flies with translational effects in humans. Now, to environmental contaminants or chemicals that are in the environment. We are exposed to myriads of these in the environment. And even though scientists usually study one chemical at a time, in the real world, it's not just one chemical that animals are exposed to. We have mixtures of this in the environment, from the home to work to industrial environment, medical facility, even water and soil. We have different chemicals in the environment. Man is exposed to some of these chemicals, and some of these chemicals have different effects in human as well as in organism. Now, in the biomedical sciences, we are always in search of the right organism, the right model organism to use to study. We have different model organisms that you can use. You can use zebrafish, you can use rodents, you can use C. elegans, you can also use salines. But what we are talking about today is how to use the fly to study the impact of environmental contaminants. So why do we now use chemical-induced Drosophila model of cancer? Why we need to do this? We try to mimic what is in the environment. If you understand what is in the environment, you can actually mimic that in the laboratory. So you mimic environmental carcinogen exposure, and then this will help to study mutation and tumor initiation. And it's also faster and cheaper than mammalian models. For example, it will take years to study some of these in mammals, and also useful for screening anti-cancer drugs. So these are different reasons why we study chemical-induced model of cancer. These are just selected carcinogens. We have ethyl methylsulfonate, benzoyl pyrene, aflatoxin, nitrosamines, sodium arsenite. Some of them are initiators. They initiate carcinogenesis. Some of them are promoters. Is that okay? So when you have different chemicals, one that is an initiator, one that is a promoter, and animals are exposed to this, it can bring about that process of carcinogenesis. And the process is to induce DNA mutation and induce genetic instability. Let's pick each of them and just talk briefly about each of them. For example, the EMS, ethyl methylsulfonate, is a laboratory mutagen. And what it does is to add archaic groups to DNA bases. It will add an archive group to DNA bases, and this will bring about the sparing during replication, and this will lead to point mutations. Apart from EMS, we also have benzoyl pyrene. It's found in cigarettes. We can convert into reactive epoxide because of the double bond in benzoyl pyrene. And because of metabolism, it will form epoxide, and epoxide are actually toxic in the system. So they can bind directly to DNA and form DNA adults and cause replication errors and mutations. So this is just the process by which benzoyl pyrene does that. We also have aflatoxin, which is found in grains and peanuts, especially when it's not well processed. It is also activated in the liver into epoxide forms, and then binds to DNA and then bring about mutation. For example, if there is mutation in tumor suppressors like the P53, you know the consequence of that. And also, it's also strongly linked to liver cancer. Then nitrosamines is also found in processed meats, tobacco, is found in tobacco, and then it also has the ability to modify DNA bases, leading to mutation. They silently form and damage DNA over time. So these are just examples of different carcinogens that we have. So in a nutshell, the big picture is the fact that the core effect of carcinogens, DNA mutations, DNA adult formation, genomic instability, cancer development. And it does not really happen overnight. To just take a process of time for it eventually to get into a full-blown cancer. And there are different patients in the hospital that the reason for the cancer may not be genetic, may not be maybe due to what is happening in the family line. It may be due to maybe exposure to some of these environmental carcinogens. And even if somebody is also, maybe if it's in the line, in the lineage, that some genes are inherited that can predispose to cancer, if the person is also exposed to environmental carcinogen, we can actually accelerate this process. So what are the routes of exposure? Maybe you want to study this and you're thinking about the appropriate route of exposure to different carcinogens. It can be via inhalation. Inhalation, for example, if the chemical is actually volatile, you can expose under controlled environment in the chamber, you expose them by inhalation. Apart from that, it can also be via injection, and then it can also be via the oral route of exposure. So what we do majorly here is to use the oral route of exposure, by mixing that with the diet, and that's what we are going to do this afternoon. We are going to see how to process the drug into a form that you mix with the diet of the flies. Now, Judith carried this out in her PhD study using N-methyl, N-nitrosourea, and benzoyl together. And then she just started the project by treating the flies and watching what happens. And after some time, she noticed that some of the flies have this rough kind of eye compared with the control that has nothing. One advantage of working with the flies is the fact that if you are working with an environmental toxicant or carcinogen, it's possible that after some time you will see some phenotypic changes compared with the controls. This was what she saw. She was not expecting this. This was what she saw, and that has actually been published. So this can be taken further to be able to look at the genes that are actually mutated as a result of exposure to this contaminant. And also, Oyibo Agogo also work on sodium arsenite, and this has also been published. And she used a plant-based approach to ameliorate the toxic effects of the carcinogen. And benzoyl and its we have also worked on this, and this has also been published in the toxicology report. You can have access to some of these papers to study them further. And the fly base is a very good resource that you can use to understand so many things about the fly. Is mainly dedicated to Drosophila. And also fly pushing. I think the soft copy is also available. We also have a copy that you can make photocopy and use. So before we go to the laboratory to experiment this aspect, because this aspect is easier for you to start with, there is no department that does not have a spectrophotometer. You can expose the flies to the chemicals and then you homogenize. We'll show you how to homogenize, how to centrifuge, pick the supernatants, and then you can start from somewhere. Is that okay? So that is easy to start once you have a spectrophotometer. Even if you don't have a spectrophotometer, you can still start your work with the fly by exposing them to whatever agent that is of interest to you. And then even if you just to look at the survivor, and then you can do a number of things. So this is easy to start with in the laboratory, especially if, for example, you don't have enough resources. So I would like to acknowledge the co-organizers of this Fly-NeuroCan workshop, Professor Ross Kagan, who is here present, Dr. Emmanuel Burati, who was here last week, and Dr. Alexander Whitwell. I appreciate all of them for joining us to make this happen. Can we give them a big hand? I also appreciate and acknowledge International ICGB, Cambridge Africa Abolaji, and the company of Abolaji that gave us the fund that made us to organize this. And Professor Kagan, I appreciate him for his support over the years. I met him in 2017. And each time you interact with Professor Kagan, you get motivated, you get inspired, you get fired up to be able to do more. Thank you very much. I also want to acknowledge H4Lab. Some of the equipment that we are using today was donated by H4Lab, and Lab Solutions also donated some consumables as well as different items. Like last week, we gave each institution present a brand new micropipette. So let's see what happens. Maybe institution present here, we also go with brand new micropipettes. So I want to acknowledge the team that we have been working together over the past years, as well as the International Advisory Board and our collaborators, and Dr. Badmos is also one of our collaborators. Thank you for your support over the years. We have visited different institutions, and different institutions have visited us, and some of them have started fly work in the university. These are just the logo of some of these universities that have the fly in the university that we are and they'll be working with, and we're open to collaboration. Sometimes we receive some attitude from some of these collaborators that we are not too pleased with, but that will not stop us from continuing to collaborate with as many that would like to collaborate with us. So thank you very much for your time and your attention. So I would like to make one or two questions, because we need to practicalize this aspect, because this aspect is easy for you to start with before you now begin to work with the different aspects, working with genetically modified flies. You may need some infrastructure like incubator and some other things to start. But with this, at room temperature, you can start with this. Maybe I can allow Dr. Alex, who was here some years ago, and how he has been able to start the Fly Lab in his institution.
Thank you, sir. I just returned from a 2 hours fellowship then when a colleague of mine gave me Professor Abolaji's phone number when I told him I'm interested in working with Fly. So I called him and he said he is trying to organize a workshop on the Fly, as if I can come, it will be good. And at that time, we pay 50,000 naira. That was what we paid then. So I came for the workshop and it was very, very impactful. I was here to be trained, but I was trained and I also trained people. So when we got there, we saw that the person that was to take us computational biology aspect was not available. So they said I should take it, and I also took that year. And since then, I've been working with the Fly. I have a very small Drosophila lab in my, although I'm using my office, I started, I'm using my office also for Drosophila, but by the grace of God, I think I'll publish a quite number of papers, even with, at least I've published 2 papers with Professor Abolaji. One was the one he displayed here with Dr. Agogo, the one he used plants to remedy arsenic-induced cancer. The other one published in scientific reports that will use a reverse straw. So the 2 have been published, and I've also tried as much as possible to publish more in my lab over there. So coming here in that particular year, I've never been a waste. So I'm only here when I saw the advert and they said we'll be having people like Professor Alex. Because Professor Alex, we have, in fact, the last FPH I applied, yeah, he actually served as my reviewer. And he wrote it, I just sent him a mail. He wrote a letter for me immediately and I submitted. And we also, applied for Abolada, like twice. So coming here has given me a very good link to all those people. As I've come here today now, I've met Professor Kagan, and I'm very sure that something will happen between us by the grace of God. Thank you so much, sir. He has been, and even apart from that, two of my undergraduate students have actually been here. They normally organize training for undergraduate students. And when they organize, they will call and give me one slot. So the first year, I sent one of my students, Bodun Damidola. He's currently in France. He won Erasmus. And the other one is Asimbi. That is, what's his name again? I call him Asimbi. That one too is he won a Japanese mesh. He's currently in Japan doing a master's. So this lab has always, really contributed to myself and my team. Thank you, sir.
Dr. Professor Mali, the comment from Professor Mali also from University of Jos.
Yeah, I first visited Professor Abolaji's lab in 2017. Spent like a month plus, and it was amazing to know that he was using fly for research. Then he has not developed very far, but he was doing very well. He has gotten a lab that time. You recently moved from your office to that lab, 2017, and I stayed there and did some work. After that, we invited him to our Africa Center of Excellence, which he trained us twice. He came 2 times, and we got seed fly from him and started our own lab. With that, I was able to go to US to visit New York University and work with Justin Blah, who introduced me to Ross. He was in Mount Sinai University, USA. I visited him there. Then recently I went to Strathclyde University and still met him at Glasgow University. So if you are here and you have not benefited, know that you are in the right place. But work is very simple to work with Drosophila and get published.